My name is Logan Swope, and today I'm going to be discussing my project. Proteomics study reveals a gender-based ribosomal inflammatory biomarker in Hepatitis C virus-induced cirrhosis. Some background information regarding Hepatitis C virus, or HCV, and hepatocellular carcinoma, or HCC. HCV impacts almost 2.4 million Americans and is a main contributor to the development of HCC. Clinical evidence suggests that premenopausal women with elevated levels of circulating estrogen are able to clear the HCV infection faster than their male counterparts, and thus show low incidence of HCC. Our studies have shown gender-based differential estrogen receptor expression in the normal liver tissue, which could contribute to protection in premenopausal women against chronic liver diseases, including HCC. It is well known that estrogen receptors regulate inflammation. The schematic below just shows the progression from a normal liver tissue to HCC, where a chronic HCV infection is a stage in this. Figure 1 on the left shows the differential expression of estrogen receptor alpha at basal level in normal liver tissues, comparing male and females. So as you can see, there is a difference in expression between female and males and a significant difference of estrogen receptor alpha. Current research has focused on ribosomal proteins as potential HCV-related cirrhosis and HCC development biomarkers. Ribosomal protein genes, or RPs, play an important role in cellular apoptosis, cell proliferation, and cancer development. Ribosomal gene, FAU, has recently been identified as an apoptosis regulatory gene, and its expression has been shown to be downregulated in ovarian, breast, and prostate cancer. There is a gap in the current knowledge of biomarkers that could be used for early detection of HCV-related cirrhosis and HCC development. Further, biomarkers that account for gender differences have not been identified. We hypothesize that chronic HCV infection may induce gender-based differential inflammation, leading to the dysregulation of RPs. The purpose of this study was to identify early cancer biomarkers in HCV-related cirrhosis by analyzing liver proteins, specifically RPs. This illustration here on the right just further shows how the dysregulation of ribosomes is known to lead to cancer. For our methods, we use de-identified and discarded normal and HCV cirrhosis liver tissues that we obtained from the National Institutes of Health Liver Tissue and Cell Distribution System. We then separated them into 4 different groups, including 10 male normals, 10 males with HCV cirrhosis, 10 female normals, and 10 females with HCV cirrhosis. Table 1 on the right just details the age range of our normal and diseased subjects in our liver tissue samples. Once we had processed the liver tissues, we sent them out to the NIH IDEA National Resource of Quantitative Proteomics Core Lab. Once the samples were received, they were reduced, alkylated, purified, and digested, and eventually mass spectrometry was formed. Proteins were then identified using encyclopedia DIA and later visualized with scaffold DIA. Proteins with an adjusted p-value of less than 0.05 and a fold change greater than 2 were considered to be significant. We were able to obtain a total of 4,443 proteins. Of those, 132 were ribosomal proteins, and wanting to break that down even further, the number of gender-based significant ribosomal proteins was 13. Figure 2 here on the left shows the altered expression of FAU observed in males with HCV-related cirrhosis when compared to females. So you can see our HCV cirrhosis male groups is downregulated significantly when compared to control males as well as HCV cirrhosis females. Table 2 just further demonstrates that with the p-values as well as a check mark for the groups that were known to be significant. FAU was significantly downregulated in HCV male liver tissues compared to both normal male and HCV female liver tissues. To our knowledge, this is the first report showing FAU as a potential gender-based ribosomal biomarker. FAU could serve as a valuable prognostic biomarker to monitor HCC disease progression and response to treatment. The role of ribosomal proteins will need further investigation. mRNA gene expression and cellular protein expression by IHC will be performed with increased sample size in order to confirm these results. We will also correlate the clinical data with FAU protein expression. Thank you for your time, and thank you to all those who help support this project.